Hello, and thank you all very much for joining today's online talk for Science Week, opening a can of worms, Parasites in the National Museum of Ireland Natural History. Perhaps you've joined us for an online event before, and if so, welcome back. If this is your first time to join an online event with us at the museum, we're delighted you could make it. My name is Geraldine, and I work in the education department at the museum. So today's broadcast is a closed YouTube event, live event, with a pre-recorded talk that will last about 15-20 minutes, and then we will go live for a Q&A. The entire event will be posted online at a later stage, so please subscribe to our YouTube channel to get notified about this as soon as it goes up. If you would like to ask questions, please use the chat feature on YouTube, and please be aware that to post a question, you will need to have a YouTube account. Before we introduce today's speaker, I just wanted to remind you that we are delighted to have visitors back at the National Museum of Ireland Natural History. We have reopened, and our capacity is limited though to the ground floor only, and this is leading to some long queues, especially during busy weekends. So if you can, a midweek visit would be less busy to avoid disappointment. And I suppose the reason why the ground floor is only open is the upper floors of the museum will remain closed for the foreseeable future because it's currently a building site, and it's facilitating ongoing investigative works into our 165-year-old roof. So keep an eye on our website and social media for the most up-to-date information about the museum. And don't forget, you can always view the upper floors on the 3D virtual visit on our website. So, while you're on the website, you might as well check out all our other talks and online resources that we have there too. So, back to why we are here today. We wanted to mark Science Week with an online talk, and I'm very excited to be learning more about parasites in the collection. So please welcome today's speaker, Zoology Curator, Dr. Amy Geraghty. Hi Amy, how are you?
Hi Ger, I'm good, how are you?
Fantastic, Amy. So thank you so much for joining us today. And I suppose let's just get straight into it because I know we've got lots to get through. So thanks a million, and we'll chat to you again for the Q&A later on. Thanks, Amy.
Will do. Hi, my name is Amy Geraghty. I'm a zoology curator in the Natural History Museum Dublin, and I'll be talking today about parasitic worms. If there's anything you find a bit squeamish, just close your eyes and pretend to be listening to a podcast or block your ears at various points. There's no obligation to see or hear all of it. I also will be starting the talk now. So the talk is Opening a Can of Worms, Parasites in the Natural History Museum. So first off, I'll be talking through what are parasites, the reasons why we study them, why we keep them in the Natural History Museum or Natural History Division, how they got there, and I'll be summarising the talk and suggesting future work or possibly reasons for you to contact me in relation to parasitic worms. With that in mind, I suppose I should tell you what a parasite is if you don't already know. Parasites, or a parasite, is an organism that lives off of or in another animal. Parasite is actually an incredibly broad term. A cuckoo laying eggs into the nest of another bird and making the other bird carry the energetic cost of raising its young is classified as a parasite. As could be a barnacle stuck onto another animal, increasing its chances or drag on it and having to expend energy to compensate for that. Parasites are a part of life. It is part of the four relationships: mutualism, where they both benefit each other; commensalism, where they benefit one side; competition, when they're both negatively affecting the other and one is negatively infected again; and parasitism. It is a very, very varied way to go through life or acquire energy. And parasitology is the scientific study of parasites. Oh, why should we study parasites and why should we have them in our natural history collections? Parasites can have huge impacts on people and ecosystems from a medical, economic, and ecological perspective. And scientists do estimate just under half of all life is parasitic. Parasites are also hard to study. most of the time they're hidden in something else, are small or cryptic, or vary in their shape, size, and appearance. So they tend to be neglected but important parts of the ecology of a system. Also, they're very interesting. They can affect their host's behavior, size, output, and are potentially a source of food for other animals in the ecosystem. So, if they have a negative effect, why not just get rid of them all? Yes, definitely there should be work done on minimizing the impact of harmful parasites. But to do that, you have to study parasites. And in the words of wisdom I have here is from an Irish parasitologist, William Campbell, who along with his colleagues, won the 2015 Nobel Prize in Physiology or Medicine. It was for their work in discovering and developing the drug ivermectin, which is an extremely effective treatment for parasitic worm infections. In his speech, William Campbell admitted he's always liked worms, always studied them, so it's a bit of a tragedy he's dedicated his life to destroying them, arguably for the better of society. But also, he demanded in his speech that it's about time parasites got a bit more respect. And I'm afraid I agree with him. And for the reasons in the following slides, I hope to cover that as well. Now, parasites in natural history in particular are a very, very important thing, especially now with the global change happening. Being able to make accurate comparisons between the state of things today to the past enables people to make accurate predictions for the future. Scientists are predicting now two very different scenarios. Either there will be a huge number or a rise in parasitic infections, or the complete opposite. So to be able to interpret that in a meaningful way or have a detailed view of it, more study of parasites is needed. And natural history museums in particular can predate the museums in our case, so before 1850, provide a really important look into the past. We also have parasites on display in the museum. So it goes to show that parasites have always been in the natural history collection and have an important status in it. There are about 50 specimens on display, and when you break them down into groups, it shakes out like this. Annelids are just a type of worm with segments. Crustaceans are your lobster, your crabs. And then flatworms are the most commonly displayed ones, and we'll be talking about these later. There's also parasitic insects or parasitic wasps that would be classified in that. Parasitic mites, which are your bedbugs, your scabies. And then nematodes, which are also known as roundworms, are on display in the museum. Next time you're in, have a look in the Irish room. Now, what are flatworms or platyhelminths? They come from the Greek of flat and worm, platy and helminth. They are soft, unsegmented creatures, and they don't have any specialized circulatory or respiratory organs. And it's roughly 3 groups: the cestodes, which are tapeworms, the trematodes, and monogenes, which are flukes. And with that, I'm going to take you through the life cycles of these 3 groups. So this is the life cycle or life history of the dog or flea tapeworm. Here we can see there is an egg in a case, or multiple eggs in a case that have come from an adult worm. They hatch, or they find a larval stage of a flea, infect the flea, and stay with it for the entire flea's life, hatch out of the flea, change into a juvenile stage of a cestode, and then, if consumed by a mammal like a dog, transform into the adult worms. Tapeworms have a really modified biology. They don't have an internal digestive system, so everything is through passive diffusion. And as you can see from the specimen in the far right close-up, the head is very small. Then there are these segments coming away from the head, and as they leave the head, they get bigger. These are maturing reproductive units. These are the parts that are passed out through the feces or through the anus of the infected mammal, and then they go on to hatch out those egg cases, and the cycle continues. Next up is the life cycle of the trematode. This again is very complicated. It requires multiple hosts to complete its life cycle and has a range of interesting shapes and behaviors that help it complete its life cycle. So The adult worm is in the digestive tract of a vertebrate, maybe a bird or a bony fish. In this case of the liver fluke, which is a very damaging pest in agriculture, it can affect a lot of cattle, so sheep and cows, and cause what's known as liver rot, because that's where the adult worm sits and can cause quite significant damage to the tissue of the liver and the bile duct. So the adult worm passes out the egg case, the egg hatches, it turns into a ciliated, free-swimming larval stage called a myrisacadia. This finds usually freshwater snail, and in that snail, it hijacks the snail's tissues, castrates it, and turns the snail into a factory producing mobile stages of the parasite. So it goes through these two kind of sausage stages known as a sporocyst and a radiate that spit out another free-living infective stage called a cercaria. Now, the cercaria is the thing that leaves the snail. We'll find a bit of vegetation, maybe some watercress, drop the tail, insist, and wait to be consumed by that cattle or that mammal. And then the cycle continues. So leaving the trematode and moving on to the speaking very briefly about the larval stage of the trematode, I just want to highlight some of the behaviors of that cercaria, so that second free-living infectious stage. It actually only lives for 24 to 48 hours afterwards. And depending on what the next host is, there's differences in their behavior and their morphology, so the shape of the infective stage. This is podocotyl. All these species are found in sea snails. This is known as a podocotyl. It's got a very small stumpy tail, and that means it's not a very active swimmer. It turns out it doesn't have to be. Its next host is a benthic crustacean, so a crawling crab or a small amphipod. That'll infect that. The next one I want to draw your attention to is the one in the middle. This is cryptocotyl, and you'll notice there's two dark dots on the cercaria of the cryptocotyl. These are eye spots. They can tell the difference between light and dark. And there's a reason for that. If you pass the shadow over a cryptocotyl, it actually moves and swims vigorously with its long tail, which you can see coming back off of it here. The next host in the life cycle of cryptocotyl is a fish. So it responds, or it's increasingly active in response to shadows and agitation. Moving on to the last one here, Echinocephala infects marine snails. So it does swim to find the next snail, but what it does is it doesn't respond to light, but what it will do is respond to gravity. So it'll always swim down. And that's more likely where you'll find a marine snail on the sea floor or on the shoreline. So all these behaviors have evolved over time to increase the likelihood of this infective stage finding its next host, even though they're incredibly short-lived. Now, last but by no means least is the life cycle of the fluke, the monogean flukes. And this is truly the one for the romantics out there, or those who have been happily married. They are 100% monogamous. They are very faithful, and we'll talk that through their life cycle. So there are the juvenile stages. So the egg is released out into the water from the adult worm. It hatches, and then it goes through a juvenile stage. Now, these are actually separate sexes. So there is a male and a female juvenile stage of this worm. If they do not find each other, they will perish. So they have to find each other. They attach via their suckers, and then they fuse together, happily and sitting on the gills of a freshwater fish, sucking its blood for many happy years together. And again, once they mature after meeting and fusing together, they will release the eggs into the water column, which hatches and continues on through the cycle. It's about the size of your fingernail. And here on the far side, you can see a very happy couple that have been preserved since 1908. So now that we've covered some of the interesting aspects of their biology and behavior, we can talk about how common are parasitic worms in the Natural History Museum's collections. They are everywhere. We have them in jars preserved in alcohol. Again, if you're squeamish, just close your eyes and I can talk you through this slide. We have them dried. through these glass microscopes, which the specimen is squished between two glass slides, and key features can be stained. There's scientific illustrations of their life cycles. We have scientific publications, and we have archive material. And just on the note of the archive material, I have to say this one is kind of one of my favorites. This is a document from the 1900s. It's a letter addressed to the keeper at the time, Dr. Scharf, and it's informing him that his specimens of parasitic worms are currently being held by customs. They either have to drain the alcohol, so he won't have to pay tax on them, and it's a back and forth between Dr. Scharf and the customs official, and it's agreed upon that the parasitic worms won't be taken out of alcohol, so they won't be damaged, and eventually he won't have to pay the taxes on it. They come to an agreement. Now, what if you want to find a specific or a singular parasitic worm? How would you go about that in the Natural History Museum and finding the needle in the haystack? Well, first of all, you check our registers. We have a paper register and an electronic database of the collections of the museum. So here I have an example in the paper register. One of the first examples we have of parasitic worms in the Natural History Museum. It is from 1892. It is the 122nd entry from 1892, and it is a collection of invertebrates and fishes. And so with that, I was able to take that number and I was able to check our electronic database. I was able to find a location code, and I was able to find the specimen in particular. So it is the specimen from 1892, the 122nd acquisition made by the year, and this is the fourth specimen of that lot. We keep as much information with natural history specimens as we can. The higher level of detail we can keep with it, the more useful it is. So here we have all the original labels with the specimen. And when we zoom in and compare with the scientific illustration of the species, we can see some of the key features have been preserved. One of the really key things is this sucker on the bottom. This is a monogean, and it has the 7 chambers on the bottom of the sucker, which we can see clearly illustrated as well on the far right. So parasites aren't lost in the Natural History Museum. If we're able to keep the paper records, the electronic records, and the preservation works, we can do a lot of nice work with that. Now, how do they get into the museum? Well, sometimes they can hitchhike. Here I have an example up of when a parasite could be in its host. So whether it's a fish or a bird, the museum or a curator accepts it. It can be frozen or go into a freezer. So first we have to thaw it. Then we put it through a chemical process called fixing or fixation, which stops the rotting or degradation of tissues. We rinse them, and then we eventually store them in a high concentration of alcohol, but we have to gradually bring up the strength of the alcohol over time to not shock the tissues, to keep it the same shape, size, and to make it comparable to how it was in life. Then we label it and store it in the collection. So in this example, I've pretended like Michael D. Higgins has found something very exciting, the first record of something in Ireland. Another way in which they come in is a researcher has been out, they've carried out a survey, they've found it, they've preserved it in alcohol or another substance already. We take it in, we accept it, we register, label, store it, and we maintain it. So say, for example, if the solution starts to decrease in volume or strength, we maintain it there. Or it can come in as a already pre-prepared glass slide. They've been preserved and stained, and we will again take these in, accept them, register, label them, and store them, in this case in a glass container or a slide store. And one example in particular of an example of taking in a donation from a scientific survey is the work of David Geraghty. He studied marine birds in Galway for 3 years, and he looked at the parasitic worms found in them. This resulted in a number of great findings and a large donation of over 1,000 glass slides to the Natural History Museum. David found over 40 new records of parasitic worms in Ireland, 15 new records for Britain, and over 40 new host records. So what that means is the parasite could have already been recorded in Ireland, but it was the first time it was recorded at these hosts. This study is really important because it kind of sets up a baseline of parasitic infection for marine birds. It was run over 3 years. The timeline is good too. And the worms, as you can see here, are well preserved and stained in the glass slide. So they're there for if anyone else wants to study them. Next up, there can also be donations from vets. This is an example of a historic donation as well. Matthew Headley Esquire was in Dunleary, and he was the Chief Veterinary Inspector of the Department of Agriculture and Technical Instruction in the late 1800s and early 1900s, and he donated numerous parasitic worms from domestic animals, so dogs, cats, cattle, things that he would have come across in his working life. They are well preserved. Some of them are still on display in the Irish Room, and they're there again with the historic record of where and when they occurred and what they were found in. Finally, there is also examples of purchases of parasitic worms in the collection. This was before David Attenborough documentaries were out on TV, or you could do a Google image search. To be able to identify and start to recognize parasitic worms, if you were a vet, a doctor, or a biologist, you had to have a reference collection. So that's what the museum collected or purchased from the case of Prague. There was numerous cestos purchased in 1888, 1908, and 1910 from a natural history collection provider from Prague, Vaclav. And then again, feel free to look away. I won't be offended. Here we have a really nice display specimen showing off a tapeworm cyst in the brain, a hydatid cyst in the brain of a sheep. And it can be seen because they've taken away the first part of the plate or the top part of the skull. And this was purchased from a French natural history collection provider for 80 francs in 1903. And moving on from that, we are still acquiring specimens. Now, we don't purchase them anymore before anyone gets too excited. But what we are trying to do is get a more representative collection in natural history, and with an extra focus on parasitic worms found in Ireland. I've highlighted a line here, and basically it emphasizes that only 5% of the parasitic worms found in Ireland are represented in our collections. And I would be very grateful if any budding biologists would like to donate voucher specimens, would get into contact with me if they would like to donate specimens. And again, there are discoveries waiting in drawers of the Natural History Museum's collection. It is impossible to be an expert on everything. So I hope this illustration shows that there is work yet to be done. So if any visiting researchers, writers, or artists want to come in and have a look at the parasitic worms in the collection of the Natural History Museum, please get in touch. We are still accepting specimens. The collections are always growing, and we're doing so with the focus on the material or getting nice work from it. And so I hope, well, just to summarize the talk, I hope I've conveyed in some way or form that parasitic worms are part of the ecosystem, are part of Ireland's natural history. They have a place in natural history collections. They're extremely varied in their biology, their behaviour, and their preparations and how they came to the museum. We do curate them and maintain them in the collections, and they are still an understudied part of natural history collections. So with that, if you have any questions in relation to what I've just covered, or if you'd like to ask anything else, please do. I'd be more than delighted to answer your questions.
Hello and welcome back, everyone. Thank you so much, Amy. I just really enjoyed your talk. Thank you so much.
Thanks, Sarah. I like talking about parasites too, so any chance.
I like hearing about them, and I have to say I really enjoyed learning about the romantic parasite.
Yeah.
You learn something new every day, and as one of our viewers said, such devotion.
Yeah, it's really rare in the natural world to have 100% monogamy, so yes. That particular monogean or flatworm is a great example for its taxa. Yeah,
Exactly. Well, and I loved your call to action at the end as well, where you were like letting people know, a huge important role of the museum is that we have these scientific collections, and we want people to use them, whether they're scientists for researching or artists or writers. So if actually they do want to get in touch with us, with you, Amy, what's the best way to do that?
We have an email that all the natural history professionals have access to, naturalhistory@museum.ie. You can find it on the website.
Exactly. And we will, if we can get a comment up, we will. I can hear a tech person busily trying to
Yeah,
get it up
I can see some of the
there.
comments coming in as well.
Fantastic. Well, listen, there's questions
Yeah.
coming in, and we are live now for our live Q&A. So,
Mhm.
let's get to it. So, the first question I think we had is about, what is the oldest parasite specimen in the collection at the museum, or in the world, maybe? What do you,
Oh,
or
in the
do you
world,
know?
I'd have to look it up. But in the
Okay.
collection, I know one of the oldest ones is the monogean flatworm that I had with the example of the text. So it goes back to 1892. However, the collection is always growing. And in some cases, we're also taking in historic specimens. And maybe if I don't get to mention it in a bit, I might just take out what possibly could be older now than 1892. I'm just working on the collection at the moment.
Yeah. Well, that's very, very old. Fantastic. Well, there are some more questions, but one that came in before was about, because parasites,
Mm-hmm.
their length. We're all kind of intrigued to know what is the longest one we might have in our collections or in the world. We like the kind of gross.
Yeah, and
And
I'm very glad you said length, because that's an important thing to know about flatworms. They can't go really big because they need passive diffusion. So there's almost a limitation on how big they can get. But in terms of length, I know I provided a picture of this. This is a historic specimen we've taken into the museum. This is a beef tapeworm.
wow.
And I'll read the label out, but it was 30 feet long before being preserved. There it is.
Perfect.
Um, yeah, Tinea sagittia. So yeah, it was 30 feet long before being preserved in alcohol. Now, the largest one in the world is a tapeworm found in blue whales, which can grow up to 90 or 130 feet, and it coils in on itself in the digestive system. So that is the longest parasitic flatworm in the world. We don't have that in the collection yet, but if anyone is working with blue whales and they find one, please let me know.
Please do. There is lots of that, like lots of research that could be done on that. Also, lots of cool events we could do around that. I'm thinking of. Yeah, fantastic. Well, there's more questions. So what I have here from Antoinette, she says, did you say that only 5% of 5% of parasites in Ireland are in MI collections, and how do you know how many parasites are in Ireland?
That's a great question. So we have records of various taxa that get published every year from National Parks and Wildlife. So they list out or make a kind of comprehensive list to circulate. So in Ireland, there have been 300 species of parasitic flatworms recorded. This is about 85 species of trematodes, 30 of monogenes, and about 55 of tapeworms or cestodes. So we know they occur in Ireland, so we can check against our records. And in our collections, we only have 5% of the recorded parasitic flatworm species in Ireland. Okay. So we're hoping if anyone again is working in the area and would like to donate reference collections or voucher specimens, please do, because it is one part of the collection I'd be very keen to get bulked up or increase. For more of a representative one, if someone wanted to come in and study parasites, they could get their eye in.
Yeah, fantastic. Okay, well, on that, do you know what is the most widespread parasite in the world?
So parasite is an interesting term. I could try and answer for flatworms. So
yeah,
parasite is just a term almost like predator. It's classifying creatures or living things depending on how they get their nutrition. So in terms of the most common parasite in the world, that's a really broad category, and I'm not sure I could answer that. But in terms of one of the most commonly studied and surveyed parasitic flatworms in Ireland is ones that, say, for example, infect commercial livestock or people. Yeah. So trematodes in tropical countries do cause a horrific disease called schistosomiasis, and that is a very common parasitic flatworm. Also in Ireland, there is a problem with the fasciola, in livestock. So those are two well studied. So we have a good idea of their distribution in Ireland. But again, everything has to be taken with a grain of salt in parasites. They're hard to find. They're usually in something else. They can be quite hard to identify. Sometimes you need to go down to the genetics level to get a confident identification of a species.
Okay, cool. Well, there's some questions here now on the preservation of them. So, from Anne, has the preservation of specimens changed since the 1800s? And do you experience degradation of older specimens? And then there's another one after that I'll continue on, but I'll let you answer those two first.
So we try to stay consistent in the Natural History Museum, and that is staying consistent with historic specimens, so we can compare like with like and aren't introducing variability in specimens. However, there is also modern health and safety and new techniques. So within reason, we try to stay as similar to the preservation techniques they had in the 1800s. We just use a lot more refined solutions, or alcoholic solutions in particular. And in terms of degradation of historic specimens, it does happen. We try to prevent it. And one of the ways in which we try to prevent it is that, say, for example, if something is preserved in an alcoholic solution, we check the strength and volume. And that tends to keep things very well. Just going back to that historic example of the monogene, you can see the key characteristics of that specimen, even though it was first acquired by the museum in 1892. Because of the way museums store things, they keep records with them, and they maintain the preservation fluid around them. You can still see it today or enjoy it today.
Brilliant. And so Gemma just adds, her question was, do you have to maintain the same alcohol percentage that the wet specimens are kept in? So they're all wet specimens, so it's the same level, is it?
So we try to. The alcohol concentrations can vary. We try to keep everything at 70%, but that's just because the strength at which inhibits microbes or inhibits degradation or rot of specimens is 50%. So we try to keep things at 70. We do surveys, but again, there's over 32,000 lots in the wet collection, and this is including 220 cabinets, which smaller specimens are kept in, 33 steel tanks that specimens are kept in, and 2 tall jar rooms, as we refer to them. So in other words, like that tapeworm I was holding, a tall jar. So we do have to be strategic how we go out and survey, because otherwise we could start that never end and there'd be 20 million other things for us.
Yeah. Okay, so we're nearly coming towards the end because we are running out of time. But we have a question here that says, do most parasitic worms start life in water?
So that is a really good question. It is hard sometimes to figure out the evolution or phylogenetics of some. Again, I'm just going to say flatworms, because parasites as a group is huge.
Yeah.
So they think, in particular, say, for example, trematodes and cestodes had a common ancestor with monogenes. So they would have moved from eating skin or epithelia of the gills of fish, and then moved on to blood, and then moved internally and lost a lot of their key characteristics, or not key characteristics, excuse me, a few bits of their morphology. So they are predominantly found in aquatic ecosystems. However, things like Phasicola are found in terrestrial systems in cattle, but yeah, it is usually quite linked to the water or aquatic ecosystems. Yeah.
Yeah. Okay, fantastic. Well, we just have time for one more. I'm sorry, everybody. There's too many questions coming in, and we're running out of time now. It's over the 30 minutes. So the one thing I wanted to ask, though, was do you have any I suppose like a resource, or where can people find out more information apart from our museum? I suppose obviously visit museum.ie, but is there anywhere else that you would recommend?
So there is a group of parasitologists in Ireland from various backgrounds like veterinary, medical, ecological, and it's called the Irish Society for Parasitology. They're actually running a photo competition at the moment for their annual calendar. I would recommend going over there. However, it's not the place to solicit or ask for medical or veterinary advice. It is just people who work in the field of parasitology that are keen to increase awareness of them and shine a light on some of their work and research.
Fantastic. Well, I love the fact that they're doing this photographic competition for their calendar, and that, you know, it's open to the public and anyone can put in their good images they have taken of parasites. Well, thank you, Amy. Just thank you so much for that fantastic talk. We very much look forward to future talks and events with you.
Okay, thanks very much, Ger. I'll talk to you.
Yeah, and just to say, guys, that we hope you enjoyed this Science
Mhm.
Week talk, and that we would like to thank everyone for watching and also participating in the live YouTube chat. So we really appreciate that, and sorry we couldn't get you all your questions. Just to say that this talk will be made public in due course, but once you have the link, you can rewatch it again and again. Also, I have, as usual, I have a small favour to ask, which is if you could please take 2 minutes to fill in the online feedback form that's being sent to you via Eventbrite. That would be fantastic. It really helps improve and inform our future events. Please keep an eye on museum.ie for further information on upcoming news about the museum and our events. And if you would like to help bring the Dead Zoo back to life, please consider leaving a donation on our website. And I know a link has been put into the chat for that as well. So that's all for now, folks. Thanks a million and goodbye. And we hope to see you again soon. Thanks, Amy. Have a good
Thanks.
day. Bye.